Okay, the outer leaflet, this is the outer part of the double membrane, consists mainly of phosphatidylcholine, followed by sphingomyelin and some glycolipids. These are sugar lipid compounds in the outer membranes, whereas the inner one is phosphatidylethanolamine and phosphatidylserine. Okay, a fifth phospholipid, phosphatidylinositol, is also localized in the inner half of the plasma membrane. So although phosphatidylinositol is a quantitatively minor membrane component, it plays an important role in cell signaling. So these are the different structures. Now, it would appear that each phospholipid can actually take up, according to where it is in the tissue, a different fatty acid. So in other words, the unsaturated fatty acid, with the one with the kink, is not fixed to being a monounsaturated fatty acid or a poly. It can be either. Depends upon what it is that's richest in diet. Now, they found this with one of the phospholipids called cardiolipin, which is rich in the heart and around the mitochondria, that if you flood the body with DHA, the cardiolipin takes up the DHA. If you flood it with oleic acid, it tends to take oleic acid in it. So nothing is totally fixed. And now they're discovering that some phospholipids naturally have saturates in them, and some, more than what they thought, have unsaturated. So again, nothing is fixed. It just depends upon the diet. So the better the oils that you are for your body type, the better your phospholipid structure is. Okay, so if you eat predominantly the wrong types of oils, if you eat predominantly trans fats or oxidized rancid fats, then you become rancid. It's pretty obvious, isn't it? Okay. So later on we'll teach you how to tell whether an oil is rancid, because that's quite important. You don't want to be taking in rancid oils. And you don't really want to take trans fats in. So the major thing is, if you do cook with oils, cook with an oil which is not the one for you, right? So your body doesn't actually take that trans fat up. You can't go too far wrong with sunflower. If you're not a red, in other words, if you're a green body type or a blue, then you're pretty safe to take up or use olive. A lot of people like coconut because it's a fully saturated or virtually fully saturated oil, and therefore it can't become a trans because there's no. But it is 10% unsaturated. People think, oh, coconut is 100% saturated. It's not. It's 90%. It's 10% unsaturated. And boy, do you know that when you use it for cooking, because it stinks the house out. And that stinking of the house is the unsaturated gone rancid, right? So coconut is all right to use, but it's not as good to cook with as people make out. Okay? As a spread, as cold, it's fine. Very good for cardiolipin, very good for the brain, all sorts of things. But it's not 100%. No oil is 100% saturated or unsaturated. You've even got a lot of saturates in fish oil. People think, oh, fish oil is great. Not that you want to cook with fish oil, but fish oil is loaded with saturated oils as well. So it's not all unsaturated. And you have got some saturated oils in vegetable oils. Okay, people again don't realize that you've got your saturates as well as your unsaturated. So cholesterol is a major membrane constituent of human cells, being present in the amount the same as in of the phospholipids. Now, this is amazing, isn't it? So it gives the turgidity is the right word. It gives the sort of intrinsic strength to the cell membranes. If you didn't have cholesterol in there, so for every molecule of phospholipid, you've got a molecule of cholesterol in there, and it holds the whole thing together. If you didn't, you'd be like an amoeba cell, and we'd be all jelly-like. Okay. So what holds us together on our cell membranes is cholesterol. Okay. Now cholesterol is therefore good, isn't it? We don't have cholesterol as a bad. It gets a bad press all the time that it's bad. Now, unfortunately, cholesterol is a sterol type of oil and can become oxidized. So in other words, it can become rancid. And what oxidizes it is, of course, reactive oxygen species, or free radicals. So when it becomes oxidized, it changes its chemical nature and becomes hardened, which makes the cell membrane less permeable. If this is the case of a red cell, it can't bend round corners anymore, so we have to get rid of it. But we can cleave out the old oxidized cholesterol with an enzyme called phospholipase and carry that oxidized cholesterol to the liver. And to carry it across, we need phosphatidylcholine. Okay, phosphatidylcholine is the great carrier. So lecithin or phosphatidylcholine is the great carrier of oxidized cholesterol to the liver. And in the liver, we then go through various conjugation, hydroxylation, conjugation pathways, and we make bile. Bile, okay? So bile is the breakdown of old cholesterol, okay? Bile is then squirted out of the liver through the tubes when we eat fats, and bile then emulsifies those fats and helps us to digest them. The trouble is that 90% of bile comes back in again, lower down in the intestine, which means you just recycle all that old stuff around and round and round. So you need something to bind your bile to pass it out. And that binding of the bile is called insoluble fibre. So the insoluble fibre, usually of cereals, will bind the bile so you don't keep recycling it. Okay? This is the great thing. If you want to get cholesterol down that way and your cholesterol is too high, you don't want to get rid of cholesterol. You want to get rid of old cholesterol, and oxidized cholesterol. So you need phosphatidylcholine, or lecithin in other words, and you need fibre. And one of the best popular ones is, of course, is oat bran. So there's this great thing. Oat bran is a wonderful absorber. The only trouble is not everybody likes oat bran. Well, it's not a matter whether you like it or not, they react to it. Wheat bran, we know a lot of people are very sensitive to, but oat bran, some people, and it blows you up after a time. You're okay for a day or two, and then you start to get big. Okay? Rice bran is much better. But rice bran is virtually, nobody's allergic to rice bran, but you've got to make sure the rice bran is organic, because they spray rice a lot, and of course, whenever you spray a cereal, the spray is going to be on the outside, which is the bran, so it must be organic. But that's the best sort of source of fibre. Pea bran, there's all sorts of ones available at the moment. But they absorb the bile and stop it from being recycled. So if you've got somebody with high cholesterol, have a look at the phosphatidyl groups, in other words, lecithin, those phospholipids, and bran. Okay, those would be the two major things to get cholesterol down. Remember, we don't want to reduce cholesterol, we want, because we need cholesterol to give the strength of the cell. What we want to do is get rid of the old or oxidized cholesterol. So we have those as markers in our kits, both cholesterol and oxidized cholesterol. So it's oxidized one that you want, because when you get the plaque build-up of cholesterol in your coronary vessels, that's oxidized cholesterol. That's not good cholesterol. It's oxidized and was damaged. Okay, so here's the structure with the unsaturated oil having the kink and the saturated having the straight part there. Now I'm going to whip through this and come back to it later on. So you flick through a few pages here, and we'll come back to that. Okay, so let's move on to hemoglobin. So if we put all those phospholipids together, in other words, the serine, the choline, the ethanolamine, the sphingolipid and the phosphatidylinositol, we call those phospholipids. So we make a composite of those 5 phospholipids that are in the cell membranes. Okay, we call it phospholipid, and it's in that little bag that Jill showed you. Okay, in there is a marker called phospholipid. So you literally put the phospholipid on. If a person strengthens to it, you need to look in that department. We need to find the right and most suitable oil for that person. All right, and then we'll come back to work out which are the best ones in that, or later on. If you don't strengthen it, don't bother to go down that route. Don't go down that route at this stage, because there's no point. Use the hemoglobin marker. Okay, if you want to know about hemoglobin, use hemoglobin. If you want to know about coenzyme Q10, use the CoQ10 marker. Okay, it's as easy as that. Those are the 3 aspects that we want to look at with hypoxia. So the hemoglobin, the quantity of oxygen carried by the saturated blood will depend upon the hemoglobin content of the red cells. Okay, I think we all know that. Okay, with a normal range of 14.5 when you have your blood test done, blood 20 mils of oxygen will combine with hemoglobin in every 100 mils. So in other words, 20% by volume there. This is the saturation of the oxygen capacity. So when we have hypoxia, what happens is we don't produce energy. And when we don't produce energy, we get a failure of the depolarization to occur. And if we're discharging glutamate and aspartate, which are excitatory neurotransmitters, into the synapse, now this will attach to the NMDA and AMPA receptors, which will allow calcium to go into the cells, and calcium then has to be pumped out, and those pumps are active, they need ATP, and if you're not producing energy, you turn to stone. Simple as that. Okay? So, excitatory neurotransmitters. Okay, you've all heard of glutamate and aspartate or aspartic acid. Okay, that's not the same as aspartame or monosodium glutamate. Okay, those are synthetic substances which can act on the same receptors and produce the same result. Okay, excitation. Now, why do we have excitatory neurotransmitters? Every sensory organ in the body has these. So we see, we hear, we taste, we smell, and we touch with excitatory neurotransmitters, okay? Because they fire and fire and fire and fire, because learning and understanding is an exciting task of seeing, hearing, tasting, touching. You can't get information into the brain otherwise. So what happens is when glutamate or aspartate opens the door, or the NMDA receptor, the gate, it leaves the door open. Okay, so lots of sodium and lots of calcium enter into the cell. Okay? And when it's done sufficient amounts, the door shuts. And the regulation of that door is by voltage channel regulators, which is zinc and magnesium, basically. So zinc and magnesium regulate the amount of input that the excitatory neurotransmitter can allow calcium in. If you're deficient in zinc or magnesium or both, you then get overexcitation, which typically we see with the hyperactive kid and things, isn't it? Okay, often they will respond very well to zinc or magnesium. Now, if it's a normal neurotransmitter like acetylcholine or noradrenaline, the door opens once and allows one atom of sodium or ion of sodium to enter, and the door shuts. Okay? Now, calcium has two pluses on it, sodium only has one, so you get double the excitation with calcium. But like sodium, calcium has to be pumped out. Once it's inside the cell, you'll turn to stone if you don't pump it out. So we have pumps which recycle it out, and those pumps require energy. That's ATP. Okay? So neurons which fire off excitatory neurotransmitters need a lot more ATP to pump it out. Otherwise the cell will accumulate calcium, and when it gets to a certain percentage, the cell dies. Okay? It's called cell apoptosis. Okay? That's how cells die. All right? So this is important because there's another excitatory neurotransmitter, which we'll see in a minute, which is why it causes so much problem with hyperactive children and with neurological diseases, neurological disorders, where the brain atrophies because it's overexcited. Okay? Now it's interesting because I don't think many people, I didn't understand this either, but now it makes total sense why it is that people with this particular problem get psychiatric and learning problems. So fundamentally, we can damage the brain from too much excitement if we don't have the energy production to regulate it. Now, at the end of the day, when we don't get enough hemoglobin or oxygen delivered to the cell, it's called an anemia. Okay, sure we all have, because you get pale and under the eyes, don't you? I think so. So anemia is a decrease in the number of red blood cells or less than normal quantity of hemoglobin in the blood. Anemia may also be diagnosed where there is a decreased oxygen binding ability of each hemoglobin molecule due to deformity or lack of numerical development, as in some other types of hemoglobin deficiency. So we're familiar with pale eyes, paleness of the skin, coldness, yellowing, shortness of breath, weakness of muscles, change in stool colours, fatigue, dizziness, fainting, low blood pressure, palpitations of the heart, chest pains, angina, heart attacks, and enlargements of the spleen. These are all things that we've just been talking about, aren't they? Low oxygen, we talked about the same things, hypoxia. So it's just a matter of the degree of them, isn't it? When you see all the symptoms of anemia, probably all of us in the room have got one or two of those. Some may have all of them. So anemia is divided up into those when we don't produce enough red cells. This is called red cell aplasia, and if we don't supply enough red cells or white cells, it's called aplastic anemia. Okay? So those two are decreases in the bone marrow, and these are mostly associated with toxins. And the major toxins in the bone marrow, which inhibit the production of the red and the white cells particularly, is toxic metals. Okay? And the heavier the metal, the harder it is to get out, and the more it gets into the bone marrow. Okay? So it's the long bones and the vertebrae which contain the bone marrow which makes ultimately our red cells. So lead is heavy, cadmium is heavy, mercury is heavy. Aluminium is not heavy. Okay? It's toxic because it inhibits certain enzymes, but it's not heavy. All right? So the word heavy metals really defines those that are higher up in the periodic table, and by definition they're heavier in weight, therefore they're heavier to get out. Okay? Now mercury is a particularly difficult. You've had all your amalgams done, or teeth done. How long ago did you have them all up?
I've only ever had one filling ever in my life.
Yes.
And so that wasn't the problem.
Okay, so we don't know whether the source of her mercury was a filling done or it's just ongoing. But as a blue person, we know that biochemically she can't shift mercury very easily. So we will see where that mercury is in a minute with you, where it's actually causing problems. Okay, we know it's in your genetic expression, your phenotype at the moment, but where is it affecting your genotype? Okay, so we'll see that we need to clear out the mercury because it's going to have an inhibitory effect because we know you've got hypoxia. Microcytic anemia, this means small cells. Okay, where the red cell is small, it's because you haven't got enough iron. And this depends really upon the hemoglobin, which is the molecule that carries the oxygen that needs the iron in it, all right? Now this is missed because they do a blood count, small number of that, but you don't really determine the hemoglobin level unless you do a hemoglobin test. And we don't know, we saw 14.6 was a good level, but we don't know what about 13 for one person, and 8 might be acceptable to another, but another one could be in real severe trouble at 13. We don't know what is normal for a person. Okay? Macrocytic anemia usually means that you've got an underdeveloped cell. So often they're larger, but not always, but they're immature. In other words, that cell hasn't matured genetically to become a proper adult for its function. And this we always associate with B12 and folic acid. We could have a hemolytic anemia because we're breaking down blood more. We could have blood loss from heavy menstrual periods or accidents and too much fluids that we've overloaded there. But when we talk about anemia, most of the time we think about a microcytic, which is a small red cell, or a macrocytic, which is a larger red cell, which is immature. But both of them end up not carrying as much oxygen, which is what the anemia is. Okay, so iron deficiency may be due to poor absorption, deficiency in a diet, very seldom because they add iron to everything these days. It could be because some other boy is taking it. Okay? Very frequently, if somebody shows one nutrient consistently, week after week that they come, think parasites. Okay? And because it means the person's not getting them. And I always think parasites when someone shows the large intestine. Could be other meridians, but certainly the large intestine, always suspect parasites. Okay, they're lurking there somewhere, you'll find them, and they're there hungry with their mouths open, just waiting for those nutrients to come down the tube so they can have them. Okay, so iron is one of the things they love. Fatty acids is another one, and Sam always talks about fatty acids. There's a really good one as well, really good one that feeds them an absolute treat. Whey. They can't wait for this stuff to come down. Whey, all these kilos that these weight people are putting into them. Whey is a fantastic way of feeding your parasite. Undigested milk proteins. Can't wait. If you want to really spread your parasites, take whey. Okay? Amazing. Okay, so hemorrhage. We could be losing blood from heavy periods, heavy menstrual periods, so we get iron deficiency. Now, if we need iron, which type of iron do we put? So this again is really, really important because what I'm just about to tell you will apply to all minerals. Okay, this is something we have only really discovered over the last 4 or 5 months, and this is really, really important because you'll find that one type of mineral will work with one patient but not on another one. Okay, so you could have a patient who strengthens to zinc, but the next patient who needs zinc needs a different type. Okay, now there's a reason for this, and this is the constitutional body type. And this is why if you learn nothing else today, you'll understand why you use one mineral for one person. Red body types use phosphates. So in the case of iron, they would use ferrous, they're all ferrous, iron, phosphate. Okay? Now, green people use chlorides, and blue people use sulfates. Okay? This is exactly the same with magnesium. It's exactly the same with sodium, and exactly the same with all minerals apart from zinc. Okay? Why is it different with zinc? is because there is no zinc phosphate which is suitable for human consumption. So instead of zinc phosphate, we use zinc picolinate. Okay, so just that would be the only exception to the rule, is you would use zinc picolinate with zinc when for red people who need phosphates. Okay. All other ones, greens need chlorides, blues need sulfates, and reds need phosphates or picolinates. All right? So it doesn't matter which one you go for. If you've got a patient with magnesium, zinc, manganese, copper, calcium, always work out which one it is. Okay? So that's why you need to have a variety sometimes of different elements to test. So if you tested, what colour are you, John?
Blue.
Blue. Okay, he's blue. Okay, so I come along and test him with zinc chloride or zinc picolinate, and he doesn't strengthen. So you say, well, he doesn't need zinc. He's desperately needs zinc. Can't taste it, can't get it anywhere else, because you're testing with the wrong type. Okay, so you must test with the right type, or a combo of all three. You know, but at least then. But it makes sense that if he needs specifically a sulfate, you give him the sulfate. Otherwise, you're giving him 2/3 of the compound that you're giving are ones that he can't use. Okay? So this will help to explain why some people will strengthen one type of compound and not another. This only applies to minerals. All right? So remember that all minerals have to be in their form that the body will take, and it goes according to their body types. In this case, it's always ferrous, which is iron with 2 pluses on it. Okay, this is the type of iron that we use at this particular stage. Now, hemoglobin and myoglobin. Hemoglobin and myoglobin contain heme. Now, my friend Igor Bondarenko, who's a Russian professor of biochemistry, he told me 7 or 8 years ago when he was working with us, he said, Chris, you should be studying about hemoglobin, heme, and porphyrins. And I said, oh, really? Why is that? He said, it's very important molecules, very important. You should understand it. So I said, oh, I'll have a look in Harper's Biochemistry, which I did. And I had a look and shut the page. And that's me, you know. And as time went on, I turned to the sort of second page after a little bit. And in the end, I didn't look at them for years and years and years until I decided I would do something on hypoxia. And the whole concept of hemoglobin became suddenly very important. And I went back to his notes and things, and he talked about heme. And I didn't see it at the time, and sometimes you don't see it at the time, why it's so important. So Igor has helped me to put this together with the chart, which is great, because I did the groundwork, and then he's made all the corrections. Okay, so I give him full marks for this. So hemoglobin and myoglobin contain heme, a cyclic tetrapyrrole. Now, there's some great names coming up, right? But you don't have to learn them, there's abbreviations. Consisting of 4 molecules of pyrrole, 1 atom of ferrous iron residing at the center. Now, I heard about this from a colleague of mine, Susie LeBray in Australia, that we used to work together 4 years ago. And she asked me then, she said, could you get some markers for me, Chris, about cryptopyrrole? And I said, oh, I'm sure I probably can, not knowing at all what she was talking about as far as what it was. And I said, well, what is it? And she said, you know, it's a breakdown molecule in the way of making hemoglobin to do with porphyrins and all these sort of stuff. So I said, yeah, I'll have a look. Anyways, it worked out. We couldn't find it. But she actually came over a couple of years ago and gave a seminar on this. And it sort of rekindled my interest in cryptopyrrole because it's involved with this hemoglobin. And I didn't really understand where it was involved. And there's a lot of wishy-washy stuff written about it. And now I can put it all together, and I'm going to share that with you so that you're very, very clear on the problems, so-called cryptopyrrole, and what is called the mauve disorder. Because I reckon at least 50% of you in the room have got mauve disorder. Okay? Now, mauve disorder covers a multitude of problems, not only including ADDH and schizophrenia and the so-called spectrum, as the Australians like to call it, but most neurological problems and down to simple problems with vision and headaches, migraine headaches particularly. And we're going to see the whole thing through, and you'll say, ah, now the sense comes to what my patients are suffering from, or yourself. Heme being the key word here. Heme-dependent enzymes. I told you I was going to tell you something which you never knew before. Heme goes on to make hemoglobin, because the globin is a series of amino acids. But heme itself is the base substance for a lot of other enzymes in the body, okay, which plays key roles in antioxidant defense, including catalase. That's the boy that gets rid of the hydrogen peroxide. So if you don't produce enough heme, you don't produce the catalase, up goes your hydrogen peroxide. Other peroxidases, these include the NADPH and other peroxidases, nitric oxide synthase enzyme, cystathione synthase. That's a long word. Anybody know where cystathione synthase is? Yes, methylation. It converts homocysteine into cysteine. Okay? So that means if cystathione cysteine enzyme doesn't function, you can't metabolize homocysteine correctly. Okay? And that means that anybody who is short of heme has got high homocysteine. Okay? And I think Patrick talked to you a lot about what all those are, and I'll give you my interpretation of that later on. Okay? But either way, it's bad news. Okay? So, if you can't produce enough heme, you can't produce cystathione synthase. Now, look on. Let's go down to here. Cytochrome P450. Okay, this is a family of enzymes that detoxify. This means if you can't produce heme, you've got defects in your detoxification system. This makes the person chemically sensitive. Okay? Some of you are chemically sensitive, I'm sure, but we've all got chemically sensitive patients. Okay? And cytochromes for the energy production. We have cytochromes called cytochrome A, B, C and cytochrome oxidase inside the mitochondrial membrane to transport the electron for making the final part of energy. That means these people are chronically tired. You remember the major symptom people go to the doctor about is fatigue. Okay? Now you can begin to get in there. So people with heme defects have fatigue. But they have inability to detoxify. They have a build-up of peroxide, which makes them more inflammation. A little bit later on, we'll see that my friend myeloperoxidase is a heme-dependent enzyme, which means they can't fire the right free radicals to kill the bugs, so they get immune system problems. There's a lot of enzymes that will come to say, now it all makes sense where the whole thing sort of fits together with it. All right, it fits together with you. So, straight away after lunch, we're going to look at the big picture. And this is going to give you something really to think about, because I think the big picture is on the front. Okay? So that's your light reading over lunch. Okay? This picture looks daunting, but in fact it becomes more and more fascinating as we get into it, because you'll see so many different conditions that you're going to meet with your patients in there. This chart is available as a laminate, which is much nicer than small. You can read this all right, but it's got a A3, double that size colour chart with two sides to it, which are available outside. And it's much nicer because you can use those as table mats to put your hot coffee on, and it doesn't matter if you spill them. Whereas if you put your hot coffee on there, you'll get a ring round there, and it doesn't look so good when you show it to patients.